It's really a pleasure to be here with you this morning, and thanks also to Jason for organising this symposium. My talk is a little bit complementary to the other speakers you've heard, in that it'll be a little bit more on the inference side, but I'm going to talk centred around the use of normative models for understanding individual differences. Hold on one second. Green button, thank you. Okay. Before I start, I'd just like to acknowledge a lot of people whose work I'm going to present today, students, mentors, and colleagues. And I'd like to talk to you about three main topics today. So first, I'll talk about how we can use normative models to parse structured variation in neuroimaging data. I would like to introduce the notion of understanding psychopathology using the science of extremes. And I'd also I'll talk you through an application where we use these kinds of models to understand rare genetic variation at the level of the individual subject at n is equal to 1. So let's get started. This framework, as I mentioned, is based on the notion of normative models, which many of you will be familiar with, but just in case you're not, the idea is very similar to the notion of pediatric growth charts, where we chart biological variation, for example, in height or weight as a function of age. Really the point being that we can use this to track development over time. And the idea in the imaging field is that we substitute these variables for things that matter to psychopathology or to understanding mental function. So learning mappings between clinically relevant variables and biological variables. Really the key feature, because this is a probabilistic model, we can use by plotting individual subjects against centiles of variation in the population, we can derive predictions, statistical predictions at the level of the individual. Now, you will probably be most familiar with normative modelling in the context of brain growth charting models, like this one from Sage Rutherford, where we chart variation in different regions of interest across the lifespan. I would like to suggest to you today that this is really only one aspect of what we can do with these kinds of models, and this notion of charting brain development or brain structure as a function of development is only really the tip of the iceberg. In fact, given that this is a general probabilistic regression model, what we can do is we can also learn mappings with other sorts of things besides structural variation, connectivity, task-related activity. And we can also use different predictive variables. So, for instance, different aspects of task design, cognition, genetics, and any kind of questionnaire data, really. Something else that we can do is that we can use these kinds of models as a common reference model to align different task variants. So, for instance, we may have a particular variant of an fMRI task which has a certain stimulus set, a certain task setup, and gives rise to a certain distribution. We might have another closely related fMRI paradigm that has perhaps slightly different variations, was acquired on a different scanner in a different population, but it taps into the same underlying cognitive process. So what we can do is we can use the normative model as a common reference model to align these different distributions to one another, such that after we transform them to centiles of variation within the population, then we can perform meaningful comparisons between different paradigms. And I'll illustrate with an example that's work from Hannah Savage and Peter Mulder. It makes use of the emotional face matching task, also known as the Hariri task, which is probably the most widely used fMRI paradigm. It's used in the Human Connectome project, in UK Biobank, among others. And the idea is the task itself is very simple. It involves matching emotions or subjects are presented with different faces of different emotional valence, and the idea is that it taps into underlying emotional circuitry. It's typically used as or very often used because it elicits very robust activity in the amygdala, but it also shows quite strong correlations often with clinically related variables, for example, anxiety. So in this particular work, what Hannah did is she assembled a very large sample using, for example, UK Biobank HCP, but also the original Hariri sample, and I'm showing the age histograms on the left-hand side. And what she did is she then applied this to a naturalistic clinical sample, which is it's called the Mindset sample. It's acquired at the Donders Institute. I'm showing here the different diagnoses of the participants in the sample. As you can see, it's very highly comorbid by design. Effectively, anyone that goes through the psychiatry clinic has the opportunity to participate in this study. So it's really a very heterogeneous sample. What we did is we then, using some kind of factor analysis model, the details are not important, we have a set of questionnaire data, and we can derive a set of transdiagnostic symptom domains that represent negative affect, cognition, social processing, and arousal. And these will be a useful endpoint for us to relate the deviations from the model to. So what Hannah did is she estimated this normative model at the voxel level. So we really want to get down to really detailed patterns of atypicality, and really tried to get as many different sources of variation accounted for as possible. So demographics, the stimulus type, even low-level parameters like the TR, the design, what subjects were instructed to do, whether they were instructed to attend to the emotions or the shapes, and site effects, and so on and so forth. When she did this, the participants in the cohort had who had a diagnosis, had a higher number of deviations. And if we map them across the brain, you can see a huge amount of individual variability. So what I'm showing here is I'm showing the overlap of individual statistic images across all patient groups. So the atypicalities at the individual subject level are spread throughout the entire task network. So we need some way of then integrating this heterogeneity to, are these patterns of atypicality predictive? And it turns out that they are. So using a multivariate regression model, in this case, a sparse canonical correlation analysis, we can predict these transdiagnostic symptom domains at the individual subject level on the basis of the patterns of deviation from the normative model. So I'll show you another example of this kind of notion. So this is work from Natalie Holtz, and it uses quite a unique cohort called the Mannheim at-risk cohort. And the idea is, can we get at early life factors that influence the development of later psychopathology? I'll just walk you through this slide. So the cohort itself is a birth cohort, and subjects were followed, they're still being followed today. And all kinds of different risk factors were obtained, for example, obstetric risk, whether the parents smoked during pregnancy, whether there's trauma, and so forth. Subjects were scanned at 2 time points, at 25 years and at 33 years. And we're going to look at a normative model that tries to get at these different factors. So what Natalie did is she estimated a voxel-wise normative model where we have these different, we predict brain structure on the basis of these different risk factors. And we can do that, first of all, in the Mars sample, and it turns out that this is the signature that we learn of adversity is very stable over time, in that it's evident in at both time points, and it was also evident in a replication sample and the imaging sample. We can start to dig into what are the different contributions of these different adversities to the predictions that are given by the model, and suffice to say that they have different patterns, influences on brain activity, brain structure rather, that are also very replicable across samples. The deviations from this model we take as once we've accounted for all these factors, these are the individual variability, the individual differences that are left, and those are in fact predictive of symptomatology, in this case anxiety symptoms. So now I'd like to talk about something else. I'd like to talk about extreme value statistics motivated by the study of climate change. And you might think that that's quite a strange thing to talk about in a brain imaging symposium, but I'd like to convince you that it's not. So I'll start by giving you a simple example. So this is, for instance, we might be interested in modeling some sort of hydrological variable. So supposing we're interested in modeling the water level in a given river system. We can define a threshold over which something bad happens, and that might be, for example, a flooding event like this one. Now, the intuition that I would like to give you that this is a really a useful way for us to model psychopathology. So we can understand psychopathology as a threshold exceedance event. And by doing the same sort of reasoning on the basis of mappings between biology and behaviour. So we can consider psychopathology as an extreme deviation from some kind of expected pattern. So if we want to, predicting the mean in these kinds of systems is not so interesting as predicting the extremes. So this is work by Charlotte Fraser. One of the problems with this approach is that considering psychopathology as an emergence, as an threshold exceedance event, one of the issues is that threshold exceedances don't occur in isolation. So if we consider, say, some kind of river system, where we have the distal causes, the genetic mutations or the environmental factors that influence psychopathology as the tributaries of this river system, we can think of psychopathology emerging at the delta, and these kinds of small, subtle causes adding up in very idiosyncratic ways to give rise to the same clinical phenotype. And this is known as the watershed model that was originally proposed by Tyrone Cannon in 2006. So can we use this as a way to understand psychopathology at the individual level? And I would like to argue that we can, and Charlotte developed a particular approach for modeling, for instance, extreme deviations across different brain regions, across different image-derived phenotypes across individuals. And we can do some kind of extreme PCA, if you like, so understanding tail dependence and how that varies across different variables. And it turns out that when we do this kind of thing, in this case in UK Biobank, we see surprisingly strong associations with many different cognitive and clinical variables. Okay. The last application I'm going to talk to you about is how we can use this kind of reasoning to understand individual variability in the genetic effects of mental disorders, or the genetic influences in mental disorders. And I'm going to take some rare genetic variants as an illustration of this point. So this is, for example, a deletion. There are many of us carry these kinds of copy number variants. This is from UK Biobank. You can see that we have these are all rare genetic variants that have been associated with cognition, for example. And you can see that, just to take one example, they're very rare. So if we're going to try to do some kind of group comparison with these kinds of genetic polymorphisms, it's going to be very difficult. But we can understand these things in the context of normative models because we can do inferences at the individual subject level, and that's exactly what we did. We have a voxel-wise normative model that shows the development of these different or cross development effectively, and we can then understand at the individual subject level how these individuals with the same genetic polymorphism actually have surprisingly different fingerprints in the brain in terms of the effects on brain structure. And I'm showing you three different examples here of individuals with the same genetic polymorphism but have effectively completely different brain patterns that are associated with it. If you're interested in hearing more about this work, I'd encourage you to come to the poster where Charlotte will be happy to talk to you about more of it in detail. So to summarize, I've shown you how normative models provide a framework for us to understand individual variation in brain-derived phenotypes. I've introduced the notion of how we can understand psychopathology as extreme deviations from an expected pattern using normative models. And I've shown you one example how using this approach and generating individual level statistics can allow us to probe the different aspects underlying psychopathology or underlying the genetic effects of rare polymorphisms. And it reveals differences that are quite surprising in magnitude. There's a lot of work that I haven't talked about. I encourage you to come and see some of the posters indicated on the slide. And I'd like to thank all my collaborators, once again, the funders, and yourselves for your attention. Thank you very much.